going to talk about cellular transport, specifically passive transport, which requires no energy. We're going to cover topics of diffusion, osmosis, and tonicity. Before we talk about cellular transport, we need to talk about the cell membrane. Every cell is going to have a plasma membrane. That plasma membrane is going to act as a selective barrier. And what that means is it's going to control what comes in and out. It's not going to let everything in, and it's also not going to block everything either. It's going to pick and choose what comes in and what goes out. To do that, it's going to have three basic components, three basic macromolecules. We're going to have some lipids here. This is our phospholipid bilayer. We're also going to have some cholesterol, another lipid that helps us with structure. We're going to have some carbs. We're going to have some sugars. We've got glycolipids, carbs attached to our phospholipid bilayer, and we've also got glycoproteins, carbs attached to our proteins. There are lots of proteins throughout the cell membrane. There's going to be channel proteins, peripheral proteins, integral proteins that go through both sides. The plasma membrane is going to have receptors for messages and be able to transmit signals to the cell's interior. So diffusion is a term that will be discussed a lot in passive transport. Diffusion is simply the movement of a solute from areas of high concentration to areas of low concentration. It does not require any energy. So we always talk about energy in biological terms in the form of ATP. Passive transport does not require any energy, and it is always going to move solutes in an area of high concentration to an area of low concentration. There are different things that can affect diffusion. One of those is distance. So it depends on the cell. Is this a unicellular organism? Is it a multicellular organism? How far does this substance have to travel? The further the distance, the more complicated it becomes, and the harder it is for something to travel across a long distance. So the shorter the distance, the easier diffusion is going to happen. What's the molecule size? So here we can see a very high concentration of a molecule, and I'm going to say that this is molecules of oxygen. We're going to have molecules of oxygen. Here's our cell membrane, and in here the cell has used up all the oxygen. So we have low to no concentration in this area here. So again, diffusion is always an area from high to low concentration, and molecules of oxygen, O2, are very small. So these guys are going to diffuse right on through the membrane, outside the cell to inside the cell. So molecule size matters. The smaller the molecule, the easier it is to get through. The bigger the molecule, the more likely it is to get trapped and stuck. Solubility is another thing that affects diffusion. Is this molecule hydrophobic, which does not like water, like a lipid, which might get through easier through that cell membrane? Or is it hydrophilic, which likes water and would have a harder time getting through the cell membrane? Temperature is another thing that can affect diffusion, going from areas of high concentration to low concentration. Living here in the southern United States, one of our favorite beverages is sweet tea. And with sweet tea, it is using our solvent of tea and water and solutes of sugar. So you add your sugar to your water. But living in the South, hopefully we have learned that it is easier to make sweet tea when that tea water is warm. The sugar will dissolve much easier in tea and give you a nice glass of sweet tea if you start with warm tea water versus ice cold tea. The sugar doesn't dissolve as well. So temperature is another factor that can affect diffusion. The warmer the temperature, the easier it is for that solute to dissolve in the substance. Another thing is gradient size. Is there a large gradient difference? Here is a perfect example where we have a very high concentration and a low concentration gradient. That's a large gradient size. That will have diffusion occurring at a higher rate than it would be if the gradient was about the same on both sides. Another thing is electrical forces. When we talk about our cells have something called resting membrane potential, where there is a charge difference between the outside of the cell and what's on the inside of the cell. That difference is negative 70 millivolts. That's an electrical difference. And different things can change that electrical force and cause it to change with the flow of different ions. So is there an electrical force difference between the outside and the inside? That can affect diffusion as well. And the last factor that can affect diffusion is membrane thickness. The thicker the membrane, the harder it will be for something to get through. The thinner the membrane, the easier it will be to get through. An example are the simple squamous cells of our epithelia that line the alveolar sacs of our lungs. Those are very, very thin cells with very thin cell membranes, so they are easier for gas like oxygen molecules or CO2 molecules to get across those barriers and get into and out of the cells. Membrane thickness is another factor that can affect diffusion. Sometimes diffusion needs a little help. If it needs a little help, it's called facilitated transport. And there's two types of proteins that can do this. There are channel proteins here in this image, and we have an open channel protein. It looks just like it sounds. Things are going to come into this open channel protein. An example with this would be water. Water has a channel protein called an aquaporin. And aquaporins can be put into the lining of cells that are lining inside of our kidneys. And when we have aquaporins in there, then water is more able to get into the cells and we absorb more water that way. With aquaporins, they're an open channel protein. Maybe the channel protein might be gated. If it's a gated channel protein, there are different things that can open that gate. Maybe it is a mechanical trigger like touch on our skin where it needs pressure. Maybe it's a ligand gated gate where it needs a chemical, some sort of ion to attach on, and then it will open. Or maybe it's voltage gated where the charge needs to be at a certain charge and then it will open that way. So our channel proteins will be open or gated, but they help facilitate diffusion. They do not need energy to open the closed gate proteins. They are just going to open with whatever trigger that they need. Carrier proteins are our last type of facilitated transport. They're similar to channels, but when the molecule binds onto the outside of this carrier protein, the carrier protein physically changes shape, and it literally carries or shuttles that solute through the protein and into the cell. It doesn't matter what the concentration of the solute is outside the cell, you're still limited by the number of carrier proteins that can actually carry or ferry those molecules across the membrane and into the cell. So we talked about diffusion, and diffusion is always going, again, from an area of high concentration to low concentration. Osmosis is a type of diffusion. Osmosis is the diffusion of water through a selectively permeable membrane. So here in this picture, we have water as our solvent. I'm going to claim this is water. The light pink stuff is water, and our little purple dots are going to be our little solutes. So here's our water, here's our solutes, and they are dissolved in water. This selectively permeable membrane right here is keeping our solutes from going across the barrier. They're trapped on either side. But what can move across the barrier is water. So on this side of the barrier, our water to solute ratio, water is very high while the solute ratio is low. But on this side of the barrier, our water to solute ratio, the water is really low in relationship to the solutes. So again, with diffusion, we want something to go from high to low. So if our water concentration is really high over here, then it's going to go across to the low side. So water can go across a selectively permeable membrane, and we can see over here that our water to solute ratio is the same on both sides now, and that is due to osmosis. Osmosis is the diffusion of water from areas of high concentration to areas of low concentration. Our last thing that we're going to talk about today is tonicity. So tonicity has to do with the amount of solute in solution compared to the cell. How does the solution compare to the cell? So we're going to say that our solute is salt in this instance. Let's change our color and go back and make it black. So we're going to say that our solutes are salt, and here's our cell, and we're going to say that its concentration is about 5%. I'm just making this up. So we're going to say this theoretical cell has 5% salt concentration within its cell. If the salt concentration within the cell is the same as the salt concentration outside the cell, if it's 5% and 5%, then what we would call that tonicity is called isotonic. In an isotonic solution, the solute concentration is the same inside versus outside. The water will be the same inside versus outside of the cell. Iso means the same. So let's talk about a different type of tonicity. What if our solute concentration in the cell was still at 5%, 5% salt concentration, but what if we drop the cell in very salty water? If the concentration of salt outside the cell is higher, say 10% versus 5%, then we would call that hypertonicity. And hyper, H-Y-P-E-R, hyper means above. It's above the level of normal. So hypertonicity means it has a higher concentration of solutes outside the cell versus inside the cell. So this cell has been put into a hypertonic solution. It's much saltier outside than it is inside. What happens to the water when it's dropped into a solution like this? So our water concentration is going to be much higher inside the cell than outside the cell. And what ends up happening, again with osmosis, the water is always going to go from an area of high concentration to low. So the water comes across the membrane and starts to leave the cell. So if you put a cell in a hypertonic solution, the water will start to leave the cell. And what happens is the cell starts to shrivel up. And what that's called is crenation. And again, that's because we put a cell with a 5% concentration of salts inside of a super concentrated hypertonic solution. And the water that's inside the cell now is leaving the cell. The cell is going to shrivel up and crenate. So what if we go back to our cell, and our cell is going to have its 5% salt concentration, and we put it in some fresh water, say at a 3% salt concentration. In this instance, the water, instead of leaving the cell, the water is much more concentrated out here. We have a higher level of water versus solutes outside the cell than inside the cell. So that means that water wants to rush into the cell. So water is going to rush into the cell to try and counteract that low concentration of water inside. And what ends up happening with this cell is that water rushes in, and it rushes in so much that the cell runs the potential of bursting or lysing. This is due to the tonicity, and instead of being hyper, which meant above, this is hypotonic. Hypotonic because 3% is less than 5%. It is below the tonicity of the cell. So let's refresh tonicity. A hypertonic solution has solute concentrations higher outside the cell versus inside the cell. The concentrations are higher outside versus inside, so the water is going to follow that, and the water is going to leave the cell, and it is going to crenate. Isotonic, iso means the same. So the water coming in is going to be the exact same as the water going out. Everything's going to stay the same, and our cell will remain a nice, healthy shape. In hypotonic, the concentration outside the cell is below what's inside the cell. So water rushes into the cell and causes the cell to swell or even rupture and burst. This is lysing of the cell. So we can see these in animal cells like the red blood cells before. We can see these in plant cells. Despite the plant having a cell wall right here, it's still going to have a cell membrane. And in a hypertonic solution, that cell membrane pulls away. It's going to crenate. It's going to pull away from the cell wall and shrivel up. In an isotonic solution, what's going in is the same as what's going out. And in a hypotonic solution, the water rushes into the cell. The cell wall gives it a little more structure, but the cell is still absorbing all of that water and swelling up. We can see healthy plant cells here that have achieved the right amount of water, and then we can see sad little plant over here, which might resemble your house plant. It resembles one of mine right now, that needs some water. This plant needs some water, so the cells have shrunk away. The little plasma membranes have shrunk away from the cell walls of this plant. It is in dire need of some water. So plant cells do this. Animal cells do this. Even unicellular organisms like this paramecium can regulate its water content. It can regulate what's going on inside of it. So hopefully we have covered the basics of passive transport. We've covered diffusion. Remember that diffusion is a substance always moving from areas of high concentration to low concentration. This does not require any energy as it is passive transport, which means no energy is required, no ATP. We've covered osmosis, which is the diffusion of water from areas of high concentration to low. And we also covered tonicity. Tonicity is what is the solute concentration that a substance is in. Is the concentration the same, isotonic? Is the concentration higher? That would be hypertonic. Is the concentration lower? That would be hypotonic.